minimize background sound levels. So please type your questions into the chat box and Dr. MacLeod will answer them at the end of the talk. Over to you.
Okay, thank you, Petra. Hopefully the audio is still good for everybody. So yeah, welcome and thanks for taking the time this morning. What Petra has asked me to do is to give a fairly high-level overview of antisense technology, and then maybe more particularly how Ionis is deploying that broad concept of that is antisense to various therapeutic settings. And I'll try and do that and focus on some examples from our drug discovery programs over the years. I know there's a bio attached to this somewhere, or my bio sketch attached to this, but just very briefly, I have been at Ionis this time for almost 12 years now. Prior to that, quite some time before that, I did a short postdoc with our founder, Stan Crook. And that's really where I kind of fell in love with the technology and the idea of antisense in its simplicity and its potential breadth of application for treatment of human disease. And I've been in touch with Ionis all along, rejoined them, as I said, 12 years ago, and have been involved in many programs, not just in the oncology space, but in the coagulation factor program, some of which I'll share with you today, in muscle diseases, in autoimmune inflammatory diseases, etc. So I'm going to really focus a fair bit on programs that I have been involved with, but others as well. And apologies, just trying. I guess I need to click this to advance. Okay, so as I mentioned, the aim of the presentation is to give an overview of antisense technology generally, drawing from examples from Ionis drug discovery programs. And what I hope to do is highlight some of the key principles within the context of those therapeutic programs. So general principles of antisense. I often like to contrast the principles of antisense with other therapeutic modalities, and particularly small molecule discovery that I also was involved with for many years in other organizations. But as many people I'm sure are aware, small molecule discovery targeted to proteins that are disease-causing or disease-modulating is a highly complex three-dimensional problem. Of course, the protein target of interest has a three-dimensional tertiary structure. It's folded, and the aim is to identify small molecules that can be synthesized and also fit very precisely into this complex three-dimensional structure. Tight interaction as well as specificity is required for the optimal therapeutic agent, and this is actually very difficult to achieve. And again, it's a highly complicated three-dimensional problem. And importantly, many of the proteins that are thought to underlie or known to underlie certain disease pathologies fall into classes that are considered either undruggable or difficult to drug. So in this particular protein structure, we see that there are some nice pockets and grooves and holes where small molecule chemical entities could conceivably bind. However, many proteins don't have such nice surfaces or enzymatic pockets and are thus difficult to drug, making that three-dimensional problem even more complex and more difficult to tackle with, for example, small molecule therapeutics. Now, just very, very, very briefly, as I think most people on this call know or remember from high school biology, is that the flow of genetic information goes from DNA coded in genes, our genes, and to mRNA, and then mRNAs are translated into proteins if they're coding RNAs. I'm going to skip over non-coding RNAs for this purpose, although Ionis is highly interested in those. And again, targeting the proteins, the disease-causing proteins with either small molecules, as I described, or even biologics, is a three-dimensional problem. However, if it were achievable to target the RNA, you could reduce this three-dimensional problem to really a one-dimensional problem, where the only thing that would be required would be the sequence of the target mRNA, and thus you could target the disease-causing protein. And this was conceptualized by Paul Zamechnik in the late 70s, where you could specifically interact with a region on a target mRNA molecule by taking advantage of the base pairing, the Watson-Crick base pairing, and designing a molecule that is encoded in the antisense orientation. So merely the complementary sequence to the target RNA, synthesize that nucleic acid molecule, and then it would specifically bind to the target mRNA or the disease-causing mRNA, and would prevent translation or lead to degradation of the target mRNA, thus depleting the disease-causing protein and modulating the disease. So that's the concept, and many companies actually in the late 80s, early 90s jumped on this concept and initiated antisense programs and or companies. Ionis Pharmaceuticals at the time, now Ionis, was only one of them. But I think Ionis differentiates itself with the kind of the unwavering commitment to the technology that we've had to solve the very difficult problems. And one of the instrumental or underpinning problems with antisense as a concept was that the nucleic acids themselves, the antisense molecules that were really just the mirror image complement to the target mRNA sequence, but still a nucleic acid, have very poor innate drug-like properties. And so what I mean by that is that these molecules are highly labile to nuclease degradation in blood and in tissues. So the phosphodiester backbone is quickly cleaved. The molecules are large, much larger than small molecules that diffuse across membranes. So these molecules do not simply diffuse across lipid bilayers of cells. These molecules accumulate in certain tissues, liver, certain cell types of the kidney, and as mentioned, there's poor cell uptake. So all of these problems had to be really systematically addressed with chemistry and other understandings to make these really non-drug-like molecules anything like drugs, or anything like entities that could behave as drugs. And chemistry, as often is the case in drug discovery, was really what led to these transformation of these molecules. And so, of course, incorporation of phosphorothioate backbone into the oligonucleotides stabilized these molecules to nucleases and made them, within the realm of molecules that could be potentially used as therapeutic. So they begun to demonstrate in vivo activities, and modifications to really pretty much every other position on the nucleotides, including the base and these 2' sugar position modifications, were really instrumental in advancing the chemistries and making these otherwise non-druggable molecules active as human therapeutics. So there's a lot of work that we're interested. There's one review here, but there are many others, and I encourage you to look into chemistry of oligonucleotides. those second generation modifications, the 2' methoxyethyl modifications, really further enabled the distribution of the molecules broadly, but also minimized the non-specific effects of those classes of molecules, particularly by decreasing interactions with several protein classes, including plasma proteins. This slide just shows you the broad distribution of second generation antisense oligos, and those in Ionis vernacular are the 2' methoxyethyl modified oligonucleotides. But when given systemically, either by IV injection or by subcutaneous injection, and now by different routes of administration, we do get broad distribution in tissues, and really pretty much all tissues, although they are tiered with respect to the amount of ASO, bulk ASO that's accumulated in various tissues upon parenteral administration. I'll touch on that a little bit more going forward. So again, the chemistry was important in optimizing the molecules themselves, but another thing that Ionis really focused on was the biology. It was originally thought that inhibition of mRNA translation was the primary mechanism of antisense-mediated inhibition of protein or translation of genetic information from RNA to protein. However, Ionis discovered that there's a nuclear enzyme, or actually a cellular enzyme, present both in the nucleus and in the cytoplasm, RNase H, that actually mediated the targeted depletion of RNA antisense duplexes. And Ionis capitalized on that finding and led to the design advances leading to what are now referred to as gapmers. And the design is shown here, where there's a 5' portion of the molecule, typically 3 to 5 nucleotides, that is modified to be either RNA-like or other variants, but not DNA-like. And these modifications lead to increased affinity for the target RNA, stability, tolerability, and other features. Those modifications can also be incorporated in the 3' region of the molecule. Again, 5 nucleotides, 3 nucleotides, and variants of those. What's required is a central portion that is DNA-like in order to support the activation and recruitment of RNase H to the now unique DNA-RNA duplex and lead to the degradation of the target RNA. That's really further illustrated here at the cellular level, where an antisense molecule that's highly stabilized by chemical modifications circulating in plasma, it actually would be plasma protein bound highly. This part of this slide is clearly wrong. These molecules don't magically diffuse across cell membranes. They're actually internalized by various mechanisms into early and early late endosomes, where they are actually released into the cytoplasm and into the nucleus. But when they then can interact with their target RNA, specifically based again by unique Watson-Crick base pairing to the target RNA. This will recruit the enzyme RNase H, or RNase H1 principally, that recognizes the now DNA RNA portion of the duplex through the DNA gap. RNase H then cleaves selectively the target mRNA molecule, and then the antisense inhibitor is then liberated and then can be recycled, or in a catalytic fashion seek out another target RNA and get rid of and then recruit RNase H and further deplete the total pool of that target RNA molecule, and then depletion of its protein. It's often interesting to remember that Most mRNA molecules are present in the nucleus, on the order of, 1,000 to several thousand copies. So it doesn't take many antisense molecules in the nucleus, given this catalytic fashion, to deplete the entire pool of 1,000 plus mRNA transcripts for any given protein. I always think that's interesting to keep in mind. But again, of course, because we're targeting the RNA, this allows us to drug very difficult protein classes, those that are considered undruggable. Sorry, I'm just reading. There's already some questions coming through, but I suppose I'm supposed to table those until the end of the talk, which I'll do. And examples of these are transcription factors like STAT3, or signaling molecules like KRAS, or unique splice variants, as I'll briefly illustrate in our prostate cancer studies, where we're targeting the androgen receptor, but unique splice variants of the androgen receptor that drive resistance to frontline therapies. And also targeting transcription factors now in other cell populations that we've only recently explored, for example, immune cells and immune cells of the tumor microenvironment. Another thing that Ionis, or ISIS back then, discovered that was of critical importance was that there are unique sites along any given mRNA that are either accessible or non-accessible to antisense-mediated depletion. That we know now holds true for RNase H-mediated degradation, but also other mechanisms of modulating RNA metabolism with antisense. So this slide just exemplifies that if you tile an mRNA, you'll find hotspots where you get significant activity with antisense oligos, but you get regions that are inactive. And this, I think, was underappreciated, broadly. And one thing that Ionis has really capitalized on to identify the best antisense molecules to bring forward into development. So now I'll very briefly describe some data from some of our Generation 2 MOGAPMER programs. And again, the chemistry is shown here. And this slide actually, I forgot to remove the graphics. So two of the graphs are hidden by the references on top of them. But the point here is that these are data, clinical data, from 4 different programs looking at Generation 2 antisense drugs, so 5-10-5 MOGAPMERs, that are targeted to previously difficult to drug factors expressed in the liver and hepatocytes more specifically. And then what's being measured is the level of that particular target factor measured in blood after increasing doses of the given antisense molecule. And as you would expect, you get dose-dependent depletion of the target protein in blood, and it's very long-lived because the half-life of these molecules is long within tissues. But I think one of the most important points is that each of these curves looks so similar in terms of their profile and their dose responsiveness and their kinetics and their onset and their duration of action. They really look almost superimposable. And if you think about it, that makes sense, because really these 4 separate drugs are chemically nearly identical in that they are 5-10-5 MOGAPMERs, 20 nucleotides in length, phosphorothioate backbones. And the only thing that really differs from these 4 different drugs is the sequence of the oligonucleotide composition. So this is, a really important point that allows many de-risking opportunities for the technology, because the pharmacokinetics in preclinical species, and monkeys, and then to man is highly translatable and predictable. The tolerability profile, the duration of action, all those things are things that are question marks, even with other therapeutic modalities going into clinical studies. Those are dramatically de-risked and predictable with the technology. And so in addition to the RNase H-mediated depletion of target RNAs, we now know that we can intervene really in many points of RNA metabolism, including modulating splicing. The RNase H-mediated degradation is shown here, but we can design oligonucleotides that don't support RNase H-mediated cleavage of varying varieties, and target these two regions, for example, that are known to be important for splicing modulation. Therefore, we can modulate splicing to lead to either new proteins or the upregulation of existing proteins through modulating the ratio of splice variants. We can also upregulate genes, and something we've been working on much more recently is the upregulation of genes by targeting regions in either the promoter regions, for example, UORFs, upstream open reading frames, or downstream modulating polyadenylation. Again, all taking advantage of antisense-mediated hybridization to target RNAs. One example of our ability to modulate splicing therapeutically comes from the program partnered with Biogen, targeting a spinal muscular atrophy. So the drug is now approved and is treating thousands, actually, of patients with spinal muscular atrophy. But the premise was that spinal muscular atrophy is caused by a mutation in the SMN1 gene, a loss of function mutation in the SMN1 gene. Just so happens that there's a pseudogene that is almost identical to the SMN1 gene, other than it has a single point mutation in an intron that regulates the splicing of exon 7. So you get the disbalance in the splice variants that are produced from the SMN2 gene. So you get the predominance of this delta 7 transcript, and that leads to a non-functional SMN protein. The hypothesis was that if we could modulate splicing of the orthologous gene, the pseudogene, and reinstate exon 7 inclusion, that would lead to a functional protein and essentially would replace the lost function of the SMN1 protein. That's shown schematically here, how in theory it would work. Several preclinical models and in disease models of spinal muscular atrophy demonstrated that intrathecal administration of these splice modulating ASOs could in fact do this. And here is just a time course showing that you can go from exon 7 exclusion that causes the disease to a shift to exon 7 inclusion and the reversion of the disease in mice. This was then quickly translated into a human clinical activity, and then to the rapid approval of Spinraza for the treatment now of all patients with spinal muscular atrophy. And for those of you who aren't aware of the story, it's really transformational. And there are now children who have been treated early who are developing completely normally, who otherwise would likely have succumbed to the disease by the age of 2 and been either on permanent ventilation or actually have expired. So Spinraza is a scientific success and quite amazing with respect to its mechanism of action, but it's also a commercial success. And last year's sales were over $2 billion, and it's been recognized in other areas as well. So really, one of the take-home messages at this point is how broadly the technology and the underlying mechanism can be applied to modulate RNA metabolism and change the disease progression. And also the capacity of the technology. Again, because we know now that we can reduce this complex three-dimensional problem to a one-dimensional problem where only the sequence information is known, and we can identify those molecules very quickly. We can go from hypothesis and genetic evidence, reducing that practically to proof of concept studies in animals very quickly, and then advance those to the human disease. Again, having all those other elements of that are risky along the drug discovery chain vastly de-risked with the technology. In any case, that has led to a pipeline at Ionis and other companies that are pursuing antisense strategies to have a very deep and full pipeline. So just maybe a little bit more on some of our generation 2 chemistries in terms of the details of how these molecules behave as drugs. Generation 2 molecules, when given systemically or locally to various tissues, really distribute broadly. When given systemically, they do distribute broadly. However, they will not cross the blood-brain barrier or cross the blood-retinal barrier. So that's an advantage, actually, for our drugs in many cases. That contrasts with small molecule inhibitors. When these molecules are distributed, however, they do accumulate in certain tissues preferentially. And those include what we call our tier 1 tissues: liver, kidney, white adipose tissue, and then on a somewhat lower level, bone marrow and immune cells. And the activity in immune cells, unfortunately, I won't have the ability to, or the time to cover today, but it's really quite striking, particularly with our latest generation chemistry. And in the oncology program, we're capitalizing on that tremendously to develop selective inhibitors of various targets in immune cells, and immune cells thought to underlie immunosuppressive states of tumor microenvironments, as well as going after autoimmune disorders. So in terms of tier 1 tissues, of course, liver is one of them, and probably not surprisingly, the area where we can practice antisense pharmacology the most robustly and selectively. So the selectivity is a key point, and the ability to ask questions, for example, of closely related family members, and which ones might be the most important in disease settings. So one example that I like to share is a program that I worked on when I rejoined Ionis now some time ago, but was on the coagulation factors. And what we did is we developed selective inhibitors to really all the coagulation factors. Here shown inhibitors that are selective for factor 11, but don't inhibit the others, selective for 7, no activity on the others, all the way down the line. And then ask the question, which of these components of the coagulation machinery are most important in pathological settings of pathological thrombosis, and which ones might have the best therapeutic window and potentially not be as important for hemostasis and regulating bleeding events. So what we found through this exercise was that factor 11, and in fact, now we know factor 12, have profound antithrombotic activity in preclinical models and in non-human primates. But really, upon significant to near complete inhibition of factor 11, we determined that this is a very safe anticoagulant as well, which was actually surprising at the time. And even when challenged with lacerations and even tooth extractions in the non-human primates that had been depleted for factor 11 levels. They really had no increased risk of bleeding. This was then brought forward into a clinical study where patients were treated with factor 11 antisense oligos prior to going into total knee replacement surgery. So that was in fact quite a trepidatious day, and even the surgeons were worried because this was a translational experiment, and we thought we could fully anticoagulate with factor 11 depletion and not increased risk of bleeding, but this was the first time in man that was actually being tested, and turned out to be true, that there was superior antithrombotic activity with the factor 11 inhibitors and no increased risk of bleeding, even in this highly challenging setting for bleeding events. I'll touch very briefly on local delivery opportunities. Ionis clearly, through the success of Spinraza and other programs in the neurodegenerative disease area, have demonstrated that local administration through intrathecal injection is broadly active in the CNS and can translate to clinical activity. But earlier stage programs targeting local delivery in the eye and by inhalation to the lung are moving forward, in addition to oral local delivery for gastrointestinal disorders. Just one slide on the local delivery in the pulmonary program that's pretty exciting. This is an asthma model where mice are challenged with house dust mite, which exposure, which leads to really all the phenotypes of severe asthma. This slide shows you that that treatment with house dust mite leads to a lot of mucus production in the epithelial cells lining both large and small airways. But the aerosol administration of oligonucleotides targeted to several factors, in this case, a transcription factor SPEDF that's involved in mucus production, but other components of epithelial cell biology in the lungs, completely inhibits and reverses the asthmatic phenotype in this disease model, where controls oligos have no effect. And now, quickly turning to an area that's now near and dear to my heart is the oncology program. And here, the generation 2.5 drugs have made real significant impacts. So the generation 2.5 chemistry advanced at Ionis are these constrained ethyl modified oligonucleotides that increase the affinity of the antisense molecule for the target RNA, but also impart several other properties that I don't have the time to go into. And the generation 2.5 chemistry has been really instrumental in getting activity in tier 2 tissues. So those tissues where there's less bulk drug delivered and thus requires a potency gain, inherent potency gain for the molecules themselves. And so these tier 2 tissues include lung, heart, fat, bone marrow, to some degree muscle, and tumors. In the cancer space, we've been collaborating broadly, both through our corporate collaboration with AstraZeneca, where we brought forward molecules targeting STAT3, KRAS, and now are advancing a molecule into the clinic targeting a transcription factor in T regulatory cells. But we have also have broad collaborations with the MD Anderson Cancer Center and now the Knight Cancer Center at up in Oregon at OHSU. And just one highlight piece of preclinical data with our very first generation 2.5 drug at Ionis, and it happened to be in the cancer program. It targets STAT3, and these data are published, but it just illustrates that systemic administration of these drugs in human tumor xenograft models, in patient-derived xenograft models, and others can really elicit antisense pharmacology. So what you're looking at here on the left is immunohistochemistry for STAT3 protein in the tumor cells of a patient-derived xenograft model. And then after treatment with the STAT3 human-specific antisense drug, you see there's dose-dependent and near complete depletion of STAT3 protein in the tumor cells. And the little bit of remaining signal and brown for STAT3 protein, that's actually mouse STAT3, and these are the immune cell infiltrates and vascular cells that are mouse in origin, and those have not been inhibited by the STAT3 inhibitor that is human-specific. Now, if we were to do the converse using mouse-specific inhibitors, we could demonstrate knockdown you would get the photonegative of this, and you would see knockdown in the tumor-associated cells of the tumor microenvironment, but not the tumor cells. It's been really useful for us. And then we were very happy when we brought STAT3 forward into human clinical trials to see monotherapy robust anti-tumor activity in the first few patients that we treated. This is just one example. This patient was had failed her previous 9 chemotherapeutic regimes, was honestly on her deathbed and being sent to hospice when she agreed to come on to the STAT3 trial. She had a very strong partial response, upwards of 80% inhibition of all her lesions about a month after treatment. And then she was stable with this level of disease for approximately 2 years upon treatment. In addition to that response, we had other very strong partial responses. This patient, again, completely refractory to all other treatments, had a very strong response to STAT3 as a monotherapy, and then went on to get a bone marrow transplant, and as we know, is cured. We saw responses in other tumor types, including hepatocellular cancer and lung cancer, shown here, very strong tumor regressions. AstraZeneca is developing the STAT3 drug now and combining it with their checkpoint inhibitor, durvalumab, and they've seen some very encouraging increases in responses in many cancers, but particularly in head and neck cancer, where it's estimated that the incorporation of STAT3 into that treatment leads to an increase in a doubling in the response to that checkpoint inhibitor durvalumab. So that's really exciting and suggests that STAT3 inhibitors and danvatirsen, as it's called now, could be broadly applied to potentiate the activity of immuno-oncology agents, in addition to having single agent activity in certain tumor settings. Now, as I briefly mentioned at the outset, there's an interesting feature of our androgen receptor inhibitor program where we can target unique splice variants that are driving disease. So without going too much into the background, I think it's probably understood that androgens and hormonal signaling in certain cancers and androgen signaling in prostate cancer is really required to drive the disease even at late stages. So much so that prostate tumors find various ways to maintain androgen receptor signaling, including amplifying the androgen receptor, mutating the androgen receptor, and generating splice variants of the androgen receptor that mediate resistance to some of the current anti-androgens. So we hypothesized that we could, because we're targeting pre-mRNA, and in this case, we're targeting intronic sequences of the androgen receptor pre-mRNA, we hypothesized that we should be able to inhibit all forms of the androgen receptor. So we screened for antisense molecules that could do that. We just demonstrated that, and then we brought these molecules, these generation 2-5 molecules targeting the androgen receptor into late-stage prostate cancer patients. We were again pleasantly surprised to see that although all patients had rising PSA levels, those are prostate-specific antigen levels that are produced by the tumor cells and driven by androgens and measured in blood. All these patients had rising PSA levels upon entry, but the majority of patients had actually stabilized or decreasing PSA levels, and some very pronounced decreasing levels, suggesting that we could in fact engage the androgen receptor in the tumor cells of prostate cancer patients that in many cases had widely disseminated metastatic disease, including bone lesions. And two of the responses were particularly interesting. They're shown here. Again, they had rising PSA levels prior to initiation of treatment with the monotherapy generation 2.5 antisense inhibitor ARRX. And after treatment, strong reductions in PSA levels. And importantly, in one patient that was characterized to have expression of the ARV7 positive mRNA species in their tumors. And these patients won't and don't respond to any therapies targeted to the androgen axis. So this was really encouraging. And also these responses correlated not only with the decrease in other surrogate markers of survival, including complete normalization of circulating tumor cells, but in fact, the patients did have prolonged survival. So we are advancing this drug in various settings, including in patients that are predetermined to express the ARV7 variant. Now, in the last few minutes, I'll just touch briefly on our chemistries and applications towards getting specific delivery of oligonucleotides to certain tissues and cell types or subcell types of those tissues. And this is our LICA strategy, and it stands for ligand conjugated antisense oligonucleotides. The furthest advanced is our LICA strategy that employs GalNAc conjugated antisense molecules. Essentially, there's a small molecule that's is an N-acetylglucosamine, essentially, and is recognized by a specific receptor or set of receptors that are expressed in hepatocytes. So by understanding that ligand receptor relationship, we can identify ligands, chemically conjugate those to our antisense drug, and have selective delivery to the tissues that uniquely express the receptors to that ligand. And we are exploiting that in many tissues outside of the liver. But this slide actually demonstrates or describes what I just said in terms of the mechanism of the targeted delivery to hepatocytes with GalNAc conjugated ASOs. So just very briefly, it's mediated through ASGPR1 and 2 and 3 receptors on the surface of hepatocytes. So you get selective targeting of the hepatocytes with the GalNAc conjugated ASOs, and then the internalization of that receptor ligand system, and then ultimately the release from the endosomes into active compartments. This approach and strategy has been brought forward into man, where the proof of concept has translated very nicely, and actually even more effectively in man, where we're getting fairly systematically approximately a 30-fold increase in the potency of GalNAc conjugated oligonucleotides compared to their parent unconjugated drug. And this really affords many things, including decreasing the doses, obviously, limiting the volume of injection, decreasing injection site reactions, and any other effects that might be class-mediated effects or otherwise, but also allows for less frequent dosing and potentially, quarterly to biannually dosing with this strategy. And as mentioned, so Ionis is extending those strategies to many other disease areas and cell types. We have demonstrated proof of concept for the LICA approach in other tissue systems, have published on some of it, but continue to explore it and make progress in other therapeutic areas. So in summary, I hope I've communicated that Antisense is a novel drug discovery platform, but that it's taken a long time to get here, and Ionis has really weathered the storm to stay at the forefront. That it's an extremely efficient technology, both from the target validation, biology exploration aspect, but then maybe even more so in the translation of those learnings to the rapid identification of the human drug for testing. Many mechanisms have been pursued and shown to be efficacious in preclinical systems and in man. So, and we hope to get many more of those moving forward into clinical testing, both in the upregulation of genes important in various therapeutic settings. And of course, we continue to advance antisense technology in additional disease areas. So the liver targeting and cardiovascular metabolic, and more recently our neurodegenerative disease programs have been concentration areas for us. But now we are advancing out of those spaces into oncology, obviously immuno-oncology, respiratory, inflammatory, autoimmune, and really any other disease setting you can think of where targeting non-druggable factors may be of benefit. And with that, I will stop and try to answer some questions. So Petra, I don't know if you can hear me, but there's a lot of questions that have come through. I'm not sure how I should take these.
Right. I can read them to you. I kind of kept copy pasting them out.
Okay, great.
Okay. And I've sorted them as well because there were some duplicates. Okay, we have the first question is from Ashutosh Dongra. Question on uptake. Do you expect unwanted degradation of ASO and unspecific proteins via endosomes? And so on.
Sorry, I don't. I heard the question. You broke up a little bit. Is it written down here?
Yes, if you just keep scrolling up, it's like right at the top.
Oh, okay.
Fairly.
Okay.
The last thing I said after the last thing I said.
Okay. Sorry, let me scroll down a second to the first question. Degradation of ASOs unspecified by endosomes. Good question. I don't think we know if there are any associations with toxicities that are coming specifically from activities or associations or interactions in endosomes. I don't believe so, because in essence that's the job of the endosomal pathway, is to essentially recycle things, and it's very fairly robust environment. So I don't feel like any of the class effects are specifically coming through endosomal interactions. Of course, when we're identifying human drugs and tool compounds, try to minimize any effects that might be mediated there. For example, pattern recognition receptor biology, but those things we capture through broad screens and really haven't drilled down as much on the mechanism to understand where intracellularly those might be happening.
The next, sorry.
Is this, can you address the issue of persistent off-target effects? Yeah, okay, sure. So, yeah, our off-target effects are always a potential risk, and more so, I would say, with shorter oligonucleotides. For example, our Gen 2/5 drugs will typically identify leads that are in the 15, 16 to 18 nucleotide length. So the potential clearly for off-target hybridization dependent or mediated effects is certainly always an issue. And so we look at that carefully, and I think everybody should look at it carefully. And of course, as you highlight, the drugs will persist for a long time in target tissues, therefore, and will mediate their on-target effects in target tissues for a prolonged period of times. Therefore, if there's potential off-targets or even known off-targets, those have to be considered and considered with respect to their duration of effects or their prolonged effects. So we consider that, and through the process of selecting both tool compounds as well as human development candidates, we carefully evaluate potential off-targets bioinformatically and other ways, and then formally evaluate both predicted and non-predicted off-targets rigorously by deep sequencing type methodologies.
I think the next question was what percentage of cytoplasmic ASOs diffuse into the nucleus?
Yeah.
Just the backbone chemistry and sugar modifications. Sorry.
Yeah, that's a fantastic question. I don't think it's precisely known, and particularly because it's difficult to image, because there's so much drug trapped in the endosomes and other structures that it's difficult to visualize due to essentially white light effects. It's difficult to visualize and therefore quantify the active drug either in the cytoplasm or in the nucleus. Some experiments where there have been microinjection studies where oligonucleotides have been injected directly into the nucleus suggest that very few molecules are actually required to deplete a given target RNA. That was done with earlier molecules and less potent molecules than we have now. So I think, honestly, I think it's going to be the answer to the question is going to be very few molecules, antisense molecules actually need to get into the nucleus to mediate target engagement and degradation or splice modulation. It's not completely known how oligos are released from endosomes and or how they traffic into the nucleus, partly because it's difficult to visualize for the reasons I described. We're looking at single photon imaging and single molecule imaging, and still there's challenges. We've done a lot of work to try to understand the pathways involved in endosomal uptake as well as release. And I would point you to the Ionis literature on those pathways and what's known about it. But I do believe that increasing endosomal release, if it can be done selectively in any way without associated toxicities, would lead to a major step change in activity for antisense drugs. Can you target the mRNA translated to the cytoplasm? The answer is yeah. The answer is yes. So RNase H is active in the cytoplasm as well as the nucleus. There may be some differences that are target by target and case by case specific, but the general answer to that is yes. Specificity. Specifically, when working with splice modulator, do you see any problems with non-specific large splice machinery? Yes and no. I would say no, principally because we try to go after sequences that have specificity. Certainly, there is a maybe somewhat more conserved sequence homology for splice donor acceptors, enhancer sites, but we have seen that we can get excellent specificity in many genes, in many exons, using splice modulating approaches. So seems to be quite specific and driven by the specificity of that particular 20-mer sequence that we're targeting. Well, PhD student, please suggestion of references to start the project. I would look up reviews from Ionis, and then maybe others. If you're specifically looking at an antisense oligo PhD project, then I would be somewhat guided by Ionis literature. And there's also a good book by Stan Crook that's called Antisense Principles, which is a good one. We consider it the Bible at Ionis. What range of ASO concentrations is recommended for okay. So what range of, I guess I should read the question, but what range of ASO concentration is recommended for in vitro testing via direct transfection? We have been moving away from transfection, particularly in functional studies, and have relied on free uptake of oligonucleotides as much as we can. And then that's somewhat cell type dependent. And certainly if you're using cell lines, it's cell line dependent in terms of the sensitivity of those cell lines to ASO uptake. And primary cells seem to be much more sensitive. But invariably we can find a cell line that we can work with and do free uptake, and the range there would be IC50s for us, or say generation 2.5 inhibitors, in the low nanomolar range for RNA and protein depletion by free uptake to the mid nanomolar range for IC50 for RNA and protein depletion. So most sensitive cells, you should get target RNA depletion in the low nanomolar range. Low to mid nanomolar range. What about off-target effects? Yeah, off-target, they fall into kind of two classes. Hybridization dependent off-targets, and then non-hybridization dependent or more class toxicities. And those can occur, and particularly hybridization mediated, can occur, and you have to carefully weed out molecules that might have propensities for off targets or known off targets due to complementarity for any reason. And then non-hybridization or more oligonucleotide class specific toxicities also can be observed, but those we would weed out during our screening processes, with essentially employ multiple in vitro as well as in vivo tolerability toxicology evaluations to select the molecules that are least prone for some of the effects of oligonucleotides that, for example, are pro-inflammatory or hepatic in nature. Example of delivery through inhalation for CNS. Oh, that's a good question. If I understand, we're potentially asking whether or not we can get CNS penetration with aerosol or intranasal delivery. And I can't comment on that, but it's interesting. I think there's potential there. we have evaluated it and are further evaluating it. But excellent question. Coronavirus, good question. There are sequences within the coronavirus that are highly conserved across the variants and are known to be required for viral replication. So there are certainly opportunities, and we've looked at it, and we think there are ASO opportunities for targeting COVID-19. I would say a potential challenge is that these are RNA viruses, and they're expressed at high levels in the cytoplasm, and again, we can't get activity there. But also typically smaller genes that are rapidly transcribed and then translated are oftentimes somewhat more resistant to antisense mediated degradation, really because the RNA dwell time in the active compartment, either in the nucleus or the cytoplasm in non-polysome fractions is potentially shorter. So challenges targeting those types of viral sequences and RNA virus sequences, but we believe it's achievable. What's the status of the STAT3 program? STAT3 drug is called danvatirsen. It's wholly owned by AstraZeneca, and they're advancing it in many phase 2 studies and are somewhat focusing on the strategy of combining it with their checkpoint inhibitor to enhance the activity of durvalumab. How many mismatches in the DNA core of the gapmer is tolerated for activity? So in the gap, as you would assume, is very sensitive to and somewhat intolerant to mismatches. One mismatch in the gap can often ablate on-target activity completely. 2 mismatches certainly would, and really 2 mismatches anywhere in the molecule often will completely ablate activity. How does it affect non-specificity? Also a great question, and something that we're characterizing very carefully at the moment. And it seems to be that you can modulate nucleotides in the gap region that can have beneficial effects on many aspects of antisense pharmacology. And so essentially enhance on-target activity, decrease off-target activity, and even potentially minimize some other ASO effects. Given the increase in potency of Gen 2.5, what is the tolerability profile, and has it been used for non-oncology indications? So the first drug in the clinic for our aerosol delivery program is a generation 2.5 drug targeting a sodium channel specifically expressed in epithelial cells, type 1, type 2 epithelial cells primarily of the lung. And so yes, generation 2.5 drugs are being employed outside of oncology indications. Other areas as well. And in addition, in our liver targeting program, where in combination with our GalNAc LICA strategy, can lead to another 10 to 20-fold increase in potency. So the totality of the gains by incorporating GalNAc and Gen 2.5 is somewhere on the order of a couple 100-fold to more than that for liver targets. Now, tolerability profile has been very good for generation 2.5 drugs, exemplified by STAT3, but also the androgen receptor inhibitor drug that I described. We went up to doses, very high doses, up to 900 milligrams, up to and above 900 milligrams per week. But 900 milligrams was highly efficacious. That's where we got the bulk of the clinical responses, and the drug was well tolerated. Do RNA three-dimensional structures predictions help to identify sequences. I would say to a limited degree, and that's and so we've relied much more so on empirical screening of the cognate RNA, ideally in its target cell population, to really understand the sensitivity of ASO, that RNA to ASOs, in as close to, for screening purposes, to the situation in the disease. So we haven't relied strongly on 3D predictions. I think they're getting better, and through incorporation of things that we have learned through sequences spanning, well, thousands and thousands, probably hundreds of thousands of oligonucleotides now. Actually, it is hundreds of thousands, and the sequence space around those data. We've used AI algorithms internally that are predicting a lot of interesting things, both on the potency and tolerability side. But we can't talk much more about that. According to one of the slides, Mo chemistry hinders non-specific interactions. However, paper by Chimeric et al., where Ionis has been involved, to suggest the opposite, since many off-target were observed for the Mo chemistry when compared to other modifications. So I would have to revisit the specific publication that you're referring to. The non-specific effects that have been very well studied for the Mo's compared to what would be generation 1 chemistry, simply phosphorothioate modified backbones, is the decrease in the pro-inflammatory activity of those generation 2 Mo gapmers. It's clear that broadly as a class, they have decreased propensity for pro-inflammatory effects, and that's been borne out into human studies with decreased rates of injection site reactions and flu-like symptoms and constitutional systems compared to earlier generation chemistries. And it definitely seems to be through decreasing protein-protein interactions and specific protein-protein interactions with the inclusion of the Mo 2' substituent. But I could revisit that paper to see if there's something that I'm missing. Can we target point mutations without affecting wild-type RNA? The answer is yes. We've done it and we've published it really in the context of the Huntington program. So there we're targeting a SNP that is associated with the mutant form of the allele and not with the wild-type form, and we can demonstrate very good selectivity. We've extended that to many other cases and demonstrated selectivity for mutant versus wild-type allele. One of the challenges with that approach is that by definition, you're restricted to that site on the target RNA. And you in essence have to be somewhat lucky that that particular site on the target RNA, 1, is accessible and 2, is sensitive to antisense mediated depletion, but it's certainly achievable. And with strategies, for example, like the LICA strategies, where we can get increased delivery to cells, and I think, for example, tumor cells and others, we will be able to then kind of, I guess I would say, routinely get activity for targeting single base differences, mutations, SNPs, and selectively without targeting the wild type allele. What are your thoughts on spherical nucleic acid platform Execure? It's those are all interesting platforms. So I should read it. So what are the thoughts on the spherical nucleic acid platform from Execure? How is Ionis differentiating from this and other competition in the CNS space? I guess those are all interesting, but in my opinion, yet clinically validated and unproven. Whereas, with our chemistries and strategies, we've clearly demonstrated that generation 2 oligos can work broadly in various therapeutic settings, including in the CNS. And so we have vastly de-risked the technology in that space for efficacy and target engagement, as well as safety and tolerability. So again, competition is always there and is a good thing, and pushes us to continue to advance the technology. So we continue to do that. How is c-Ed oligo different than other LNAs? So c-Ed and the various variants that we evaluated prior to selecting our generation 2.5 chemistry, so the constrained ethyl modification, we profiled it, very carefully compared to other entities, other substitutions, including LNAs. And there are a vast number of differences. Probably they come down to subtle differences in protein binding and the interactions with those proteins and RNase H and the RNase H complex and target degradation. But the bottom line is that the therapeutic window for LNAs is significantly narrower compared to c-Eds. I think that's due to a number of factors. Again, specificity at the level of cleavage, so through RNase H versus on target versus off target, and other non-hybridization dependent off targets that the LNAs seem to universally have compared to c-Ed modifications. Okay, how do ASOs cross the plasma membrane? ASOs are internalized by interactions with various cell surface proteins, essentially binding to the surface on various different cell types of interest, and then engaging or initiating the endosomal internalization of the ASOs. And that then leads to endosomal processing of the ASOs, but because of their chemical stability, they can survive the endosomal processing from early to mid to late endosomes and then to lysosomes, and escape from that endosomal maturation at some point along the way. We don't know exactly where the bulk of the escape happens, but it seems to be along the process of early to late endosomal maturation before they get to the lysosomes. Are ASOs immunogenic and generate ADA? So ASOs have the potential to be immunogenic, and that has to be minimized and can be minimized, again, through the selection process of, for example, the human drug. Oligonucleotides have shown that they can lead to anti-drug antibodies. To date, none of those have been high titer, and none of those have been neutralizing. So that's very much a positive, similar to many protein therapeutics, and actually superior to many protein therapeutics, where neutralizing anti-drug antibodies seem to be somewhat more commonplace. So they can be seen, and certainly, when doses are lowered, for example, through LICA approaches, those incidence of ADAs is diminished.
Right. So I think we need to finish this up now. Thank you very much, Robert Dr. MacLeod, for this talk, and thank you everyone for attending. Please check out the OTS website for our other upcoming and past webinars. In particular, for those who've been listening to the questions, there are webinars by Steve Dowdy on endosomal escape and Art Creek on immunogenicity of oligos. You can also follow us on social media at OT Society on Twitter. And I hope to see you all at the next OTS annual meeting in September in Montreal.
And Petra, I would just add, if anybody has follow-up questions or interest, feel free to reach out to me by email. I don't know if my email is included here somewhere, but I'm happy to further conversations.
Yeah, or if people want to email, and then I can forward it. Email OTS and I can forward it.
Great.
I wouldn't want to give out your email online like this. Thank you very much for doing this.
Very welcome.
Bye, everyone.